So now I'd like to introduce Luc Jaeger. He's a professor in the Department of Chemistry and Biochemistry at UC Santa Barbara. He received his PhD in Structural Biochemistry and Biophysics from the University Louis Pasteur of Strasbourg in 1993. After postdoctoral studies at Scripps, he worked as a CNRS research scientist at the Institut de Biologie Moléculaire et Cellulaire in Strasbourg before joining UC Santa Barbara, where his research focuses on RNA folding and evolution, nucleic acid-based bionanotechnology and biomaterials, and emergence of complexity in living systems. So today, he'll talk to us about the origin of life on Earth, how the tree of life is rooted in matter.
Thank you. So, thank you very much to the organizer, and especially to Stephen Barr, to give me the opportunity to present some of the work that we have been carried out at UCSB, but also what I will do here is more on giving you an overview of what's happening in the field. So, as today it's the Feast of the Holy Trinity, I really think important to mention Saint Bonaventure, who in multiple writings emphasized that creation reflects the Holy Trinity. So how the Holy Trinity is reflected in the creatures according to Saint Bonaventure? Well, in being in matter, it's reflected as unity, truth, and goodness. In being in intelligence, in memory, intellect, and will. And in being in eternal art, faith, hope, and charity. So what I will do today is in fact trying to give you a meditation on what is life, on what we have been able to understand of what life is through science, but also through the eyes of a Christian Catholic. Very importantly, when one sets out to seek wisdom by a curious investigation of creatures, then wisdom strays farther away. These words of Saint Bonaventure are very important to keep in mind, as most of the time, as I've witnessed science, it's not really the spirit that animates science. It's more a spirit of curiosity, trying to extract by all means knowledge. While I do believe the Catholic approach would be to learn how to contemplate and to let, in fact, this knowledge coming to you as it is a gift from God. So science should never be an end in itself, but a means to glorify God, the Creator. As such, we need to learn, as God contemplates the earth, to contemplate the works of the creation, finding God in it. So today, my talk is essentially focusing on the origin of life on Earth, of course, from a perspective that is scientific. But you will see in many ways that through this meditation, what's reflected is also this Trinitarian vision of our God. So what do we know about the evolution of life on Earth? Well, here is displayed the abiogenic scenario for the evolution of life on Earth according to a fresco that is displayed at NASA Ames. I found it quite beautiful and meaningful. It's basically exemplifying the fact that us, as human beings, are those able to look at the stars, look toward the future, but also somehow looking at our past, trying to understand from where we are coming. And this abiogenic scenario entails the notion that as part of this universe, well, we are in fact also in part rooted in this universe producing eventually molecules that could have complexified, self-assembled, created unicellular organism, themselves complexifying to give a multicellular organism. All this leading to a process of evolution to who we are, the observer. but also not only observers, very much actor within this process. So first of all, let's have a look at the ingredients of life and what we can learn from science. When we're looking at the ingredients of life, well, basically what we're dealing with is a chemistry that is remarkable, that is a chemistry of carbon, well, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, but mainly what we're calling organic chemistry. A chemistry that is rooted, in fact, in some of the most simple atoms produced by the stars. We have seen that how basically hydrogen and helium came to be through the process of nucleosynthesis. The reality is that if you're looking at what are the most abundant atoms in this universe, beside hydrogen and helium, well, what you're finding is that carbon, nitrogen, oxygen are really among the most significant abundant atoms. And these atoms, because they are carbon, nitrogen, or oxygen, they have remarkable properties, leading them to react with one another. And this product of chemical reaction leading, in fact, to molecules, organic molecules, and these organic molecules, interestingly, science has been able to investigate them, to analyze them, to look at them in multiple environments. And as we looking at these molecules, organic molecules also, the question has been, in fact, how they came to be at the first place. And so this abiogenic scenario for the beginning of organic life on Earth, now I want to say that it's not just one unique scenario. It's in fact a scenario that takes advantage of all the knowledge that can be garnered at the present time through science. And what is interesting is that as we're considering the chemical evolution on Earth, chemical evolution of these basic organic molecules, in fact, it's really an evolution that has taken advantage of multiple contexts. And we will see that according to some of these contexts, well, there has been also a possibility to really identify some of these building blocks that enter into the composition of living organisms. So when we're looking at this abiogenic scenario, there's different possibilities. Well, organic molecules, based on the presence of small inorganic compound, could basically be generated eventually in the atmosphere through atmospheric chemistry. But they can also come from space, eventually through comets, meteorites. And what are, after all, the composition of the nucleoclouds that separate stars and that generate, in fact, stars? What about planet Earth? Is there any place on planet Earth that could basically promote efficiently the synthesis of organic molecule? What about the ocean? Is the ocean a good place for having organic molecules coming to be? Is basically is this process taking advantage of minerals? All these basically have been proposed as possible context or leading to the origination of organic molecules. And we will see that in many ways, each of these proposals are leading to the formation, are supporting the formation of organic compounds on planet Earth and in this universe. So the first experiment that was done quite a long time ago, in 1953, by Stanley Miller, was basically to check whether it was possible to generate upon in a setting that was mimicking, in fact, the condition of a prebiotic atmosphere on Earth, his experiments was to verify that eventually organic compounds entering into the composition of life, namely amino acid or other compounds, could basically be generated through by exciting basically with an electric spark a gas mixture, looking at the way this gas mixture formed essentially of ammonia, methane, water, could potentially lead to the formation of the building blocks of life. So what are the building blocks of life? There are amino acids, there's the bases that enter into composition of DNA and RNA, there's the sugar that also enters in the composition of acid nucleic, and of very important polysaccharides that could support life. It's basically also lipids. All these are the organic compounds essential for having life. So the experiments of Stanley Miller, remarkably in 1953, led to the demonstration that something like 10 amino acids entering into the composition of living organism, proteins, could be generated. At that time, the hype was to say the origin of life problems have been solved. But of course, we're much more than merely materials. It's important to look at the matter we're made of, but of course, we're still far from life. But this was really one of the first experiments showing that, after all, organic compounds could be generated quite easily. Now, analysis of meteorites and comets have also led to the discovery of organic compounds present at the level of these comets and meteorites. In this case, it's a paper describing the analysis of carbonaceous meteorites that showed that they contained a quite wide range of extraterrestrial nucleobases. Now, what are these nucleobases? Well, there are different types of nucleobases, but also those including adenine and guanine, nucleobases that enter into the composition of DNA and RNA. So now, one of the latest investigation in this area was basically the one on the carbonaceous asteroid Ryugu that show that after all, uracil could be also identified at the level of pristine samples coming straight from this asteroid. So we all the time have a doubt when we're doing experiments on meteorites that fall on Earth. Maybe there's a problem of contamination here through the fact that these meteorites enter into the context of Earth. But the reality is that in this case, here again, we're validating, science validates the fact that these bases could be generated in space. What about nuclear, the clouds, the interstellar clouds? In fact, there's a great deal of analysis that have been performed by radio spectrophotometry looking at the composition of these clouds, interstellar clouds, this composition revealing that there was presence of silicates, water, and also organic compounds. In fact, the largest amount of ethanol is in space, at the level of the interstellar clouds. Experiments have been performed on conditions mimicking, in fact, what we think happen and what is present at the level of these interstellar clouds. Interstellar clouds, in fact, they are formed of cosmic dust particles that are basically particles of grains of silicate that present, in fact, micro nano environment within which chemistry happens. And scientists mimicking these conditions in the laboratory found out, after all, that under ultraviolet irradiation, ribose entering into the composition of RNA and related sugars could be found, produced through this type of process, mimicking the condition of interstellar clouds, nanoparticles. So these particles, in fact, are grains of ice. Silicate covered by ice, and chemistry occur in the ice layer surrounding these particles. So what about now organic compounds emerging in a kind of prebiotic soup? Well, the state of the art at the present time in chemistry has demonstrated that there's possible common origins of RNA, proteins, and lipid precursors by mimicking conditions of a protometabolism, for instance, in this case here, a cyanosulfidic protometabolism. All these compounds here that you see, well, from very small compounds such as acetyl. So there's hydrogen cyanide and acetaldehyde present at the origin within the soup. In fact, it's possible to imagine chemical reaction that, in a stepwise fashion, could lead to the production of a full-fledged RNA nucleotide building block. So it's not here merely, and when I'm saying stepwise, in fact, it's more taking advantage of a systemic type of chemistry where you're realizing that this ensemble of reactions could, in fact, form a network of chemical reaction leading to the full-fledged production of an RNA nucleotide building block. In the past, it was thought that, well, you need first to form the base, then you need to form the sugar, then you need to add a phosphate. Here, everything can happen within the context of networks of chemical reaction. So RNA building blocks could be formed. This was thought for a long time that it was very difficult to do. Amino acid could be formed, and even the precursor of lipids. So to go from this type of chemical networks of reaction to the possible emergence of a chemical network that is similar to our metabolism, of course, there's still quite a bit of challenges to be overcome to understand how all this happen. But the reality of all these experiments are clearly demonstrating that when we're talking about building blocks entering into the composition of living organisms, they're readily accessible based on the chemistry that takes place within this universe and in different contexts. Now, when we're talking about building blocks, well, we still need to go further towards really the macromolecules that are entering into the composition of living matter. When we're talking about what we are made of, yes, we're made of these building blocks, amino acid, sugar, and nucleotides, but the reality is that these building blocks, they are linked to one another, creating biopolymers. Building blocks, it's a bead, but here it is, the biopolymer. And when we're talking about the biopolymer, well, it is truly, because of the nature of the building block, an information that is able to fold into defined shape according to the building blocks that enter into this polymer. So how easy it is to obtain polymers, after all, once you're having activated nucleotide building blocks? I'm focusing here on RNA. RNA that was at one stage thought to be very difficult to synthesize. The reality is that if you're taking activated monomer of entering activated monomer nucleotides, if you're putting this activated nucleotide monomer in presence of some kind of clay, in fact, what you're ending up after several hours is a production of multimers. And here, conditions are such that, well, after 3 days, you end up with multimers that could be up to 40 nucleotides long. Possibility to generate libraries of all these polymers. More recently, in the lab of Steve Benner, what they did is that they took some prebiotic rock glasses, minerals, olivine, different minerals, and they just check what happened if I'm taking these minerals and covering them with nucleotides. Well, after some hours, well, days, what you're observing is production of long chain of RNA. Well, to tell you the truth, this is not solving per se the scenario that took place on planet Earth, but it's really telling us that as far as it concerns the production of these biopolymers, conditions could be found and tuned still, so that we could definitely imagine this polymer not being such a hurdle to come up to life, well, to reality. As I said, RNA, polypeptide chains, DNA, they are informational polymers. Now, my work has been essentially focusing on RNA, and I think RNA is really a very interesting biopolymer, as, after all, like proteins, it can fold into defined shapes that could carry eventually functions such as catalysis, recognition of other molecules, but also like DNA can be used as a support of the genetic information. In fact, when we're looking at a molecule of RNA, we're looking truly as a biopolymer, an informational biopolymer that has multiple layers of expression of an information. It has a tremendous potential, that's what it means. And how is this potential taking shape in matter? Well, essentially, because of the ability of this polymer, organic compound, to generate non-covalent interactions. How does it fold? It folds because there's multiple weak interactions that can take place at the level of the polymer itself, but it's also because of this weak interaction and this ability to interact with other molecules that you're dealing with a molecule, a type of medium that has catalytic function, recognition function, ability to enter into cognition with the medium that surrounds it. So, hydrogen bondings, pi stacking, coordination with metals, electrostatic interaction. All these interactions are taking place in an even way within RNA molecules. And even way within the molecule, but also in interaction with the medium. For example, water is structured around these molecules, showing here the interplay between water, aqueous medium, that is in fact the medium for life on planet Earth, and many other chemicals that are surrounding this molecule. So it's not only water, but it's water and all what is available out of the chemistry on Earth. Talking about functions, do we need very long polymer to have a function at the level of an RNA molecule? Well, the reality is that no less than 5 nucleotides is enough for having chemical reactions catalyzed by this small little polymer, leading to eventually translational products. We will see a little bit more what it is, but basically here an RNA of 5 nucleotides is able to recognize a substrate of 4, leading in fact to the formation of small dipeptide, tripeptide. Here, the interplay between amino acid and nucleotides leading to the production of not only RNA as we have seen, but here, RNA helping the synthesis of peptides. So the meditation that we're entering now is to look truly at what is life. We have seen how the compound and ingredient can come, but the reality is that what is life? So let's have a look at the meaning of life from three different perspectives. One is Zoe. Zoe, in Greek, encompasses, for me, the notion of processes, general processes that pertain to life, the essence of life. In fact, in the Bible, it is what is often used for describing true life by Jesus Christ. Zoe. So, interestingly, this is a life that encompasses the unicity of the fundamental processes of life. Recently, this is an experiment performed in 2016, it was published in 2016, that basically looked at the way scientists could define minimal living organism, and the lab of Craig Venter engineered basically an organism, Mycoplasma mycoides, which is one of the smallest living organisms on planet Earth. Taking advantage of this natural organism for defining a version of it that had a minimal genome, reducing the genome of this organism. So they ended up basically by genetic engineering with an organism that had no more than 473 genes, totalizing basically 500,000 of base pairs at the level of the DNA genome. This organism being able to grow and multiply. Interestingly, when you're looking at these genes that are essential for life, half of them basically are involved in the expression of the genetic information and also in the conservation of this genetic information by mechanism of repair. When we're looking at this cell, in fact, what is a cell? A cell is a bag, a lipidic bag, within which you have multiple, basically, organic polymers, compounds, at the level of which the metabolism is performed. Well, the reality is that when you're looking at a cell, you can really here realize how complex it is. Even it's composed of no more than 473 genes. Multiple compounds are basically packed within this cell, interact with one another according to some chemical reaction, all this basically leading this organism to be able to take in food from its environment, produce its own polymer, and multiply from one mother cell, giving two daughter cells, etc., etc., etc. So what are the chemical reaction taking place here? Well, that's essentially centered around informational pathways that lead, in fact, an information encoded at the level of the DNA to be transferred to RNA that basically could be seen as a transient copy of some pieces of the DNA. RNA that is then translated into proteins. So these operations are absolutely necessary for life as we know it at the present time. And this entailing DNA replication, well, the production of RNA from DNA by transcription, translation of RNA into protein, but also, interestingly, there's a possibility to have RNA reverse transcribed into DNA and RNA replicated itself through protein-mediated reactions. Now, this is how the central dogma is typically represented, but the reality of it is that it's a hypercycle network. That's what it is. And this is a much better way of depicting all these operations that takes place for basically operating the cell. It's in fact a cyclic dynamical chemical network of reaction with multiple feedback loops. And this is truly responsible for cell multiplication and reproduction. Now, what is a key element, in fact, after all, as we're looking at this process? In fact, one of the key elements, of course, is the ability of RNA to generate function. Now, RNA could be itself a function, but it also, as carrier of the genetic information, it could be translated into proteins. And this is operated by the ribosome. The ribosome is the largest machinery. It's an incredible machinery that is present in all living organisms on Earth. It's totalizing at least 150 elements just for building it up. But this is what defines life. It's the key of life. This is why we can say you have a cell. When you're finding a ribosome in you basically, the ribosome is related to a living organism immediately. And this is what is able to process the universal genetic code of all living organisms. Again here, we're talking about a machinery that is expressing, that is able to decode an information at the level of a DNA or RNA into a protein, and the coding system that is used by this operation is universal among all living organisms. Meaning that if you're looking at a bacteria or a human being, same type of genetic code. Talking about the complexity of the system, well, to tell you the truth, now we have a great deal of understanding how it works, but it's definitely not simple. And that is basically what characterizes life on planet Earth. So how can we come from mere ingredients to the realization of a system as complex as that? That is really the great challenge at the present time in biology. So looking at the ribosome, what is interesting is that the ribosome is formed of RNA associated with proteins. And it was found that it's the RNA that is responsible for the chemical reaction leading to the formation of all modern proteins. Basically, all the proteins that you have in your body, they're made by an RNA. So, RNA seems really to be one of these molecules, fundamental molecule, on which life could be based. And this led, basically, scientists to propose that, after all, at some times during the evolution of life on Earth, life was essentially controlled by RNA biopolymers, with genetic continuity assured by the replication of RNA on peptides synthesized by RNA. We know that RNA synthesizes proteins. We just have to look at how it's happening now. The reality is that the problem might be a little bit more complex when we're looking at the ability of having an RNA replicated into an RNA by an RNA. Now, what is interesting, so, is that as we're looking at the way living organisms presently on Earth function, well, that system that is depending on RNA, proteins, and DNA, it's possible to imagine pathways that could have basically from a simpler system based only on RNA and peptide, a system that might have then evolved through time into what we know. It's a kind of retrogression type of work here that could be seen. And interestingly, it's possible to look at an increase of complexity in life driven forward if you have an RNA that is able to synthesize an RNA, and that this RNA is also able to synthesize peptide. Peptides could increase its fidelity of fabricating RNA. The RNA is able to fabricate other RNAs and with higher fidelity. This leading to genomes that are longer, genomes that could be RNA genomes, but that could basically carry more genetic information, leading to new functions that could increase the fidelity of replication of this RNA, leading to longer genomes, etc., etc. In fact, what you have here is a movement that could be initiated from an RNA replicase, that is an RNA, that has a potential also to interact with peptide and synthesize peptides for pushing the system forward. And this is, in fact, what we think probably has been very important for the origin of life. In fact, a system, a mini genome that, in fact, encode a RNA replicase, a protoribosome, regulated by riboswitches, a system that basically was pushed forward from its ability to replicate RNA and interact with peptides to a system inventing DNA and modern proteins. How is it possible to imagine this type of thing? Well, here we need to go away from this mere Darwinian evolution type of process. There's an interesting paper published in 2006 by Carl Woese, who is really one of the pioneers to understand evolutionary process at the level of the ribosome, that basically proposed a collective evolution for the emergence of the genetic code. It's a very interesting idea, because what it is saying is that the refinement and selection of innovation sharing protocols at the level of RNA eventually, basically a sharing protocols that encodes a protoribosome could have resulted from non-Darwinian mechanism of exchange of genetic materials within a communal population of cells. So what does it mean? It means that if you're looking at a population of cell, this population of cell, it's not like each cell lives for itself. There's no selfishness here. They're all living for one another. As they are living for one another, they're exchanging genetic information between one another. As they're exchanging genetic information, each cell has to understand the genetic information that is sent to it, that is able to get from the other cell. This leading, in fact, to a driving force towards the emergence of a unique genetic code. You have here different lineage of cells. They're exchanging horizontally information. And as they're exchanging genetic information, this exchange of genetic information starts to be the driving force for evolving a new language that is common to all the cells. A genetic code. The genetic code here resulting from communication and exchange. Of course, all this move through time in a vertical fashion, leading also to the divergence of some of these organisms. But the reality is that from within, they are based on the same language. So, this is non-Darwinian. It's Darwinian and non-Darwinian. It's Lamarckian and Darwinian. Here we can toss in the bin all these ideas of selfish genes. And that is basically what we need to teach to our students, because clearly, instead using ideas of selfishness here, its cooperation and collaboration. Now, it's very important to be able to demonstrate that an RNA might have the capability to synthesize itself. And there has been incredible technologies that have been developed in the laboratory by the lab of Jack Szostak, Larry Gordon, Jerry Joyce. And basically this process of in vitro selection and evolution in the test tube allow from a large population of sequences to have a selection taking place to isolate, amplify selectively some of the sequences that are fulfilling a certain selection criteria. And there has been, in fact, in vitro selection and evolution experiments performed for looking at RNA able to synthesize RNA. So over a period of almost 3 decades, scientists have been able basically to realize great progress, demonstrating that after all, an RNA can synthesize an RNA. And in fact, here you have a product that is 206 nucleotides that has been synthesized by an RNA of the same length. So basically, an RNA can synthesize another RNA of its length. More experiments have been performed, and this is the last paper of a long series of papers that demonstrate that after all, RNA polymerization can basically be performed in a quite processive fashion. So we're not here matching the activity of proteins that synthesize RNA, but we are really at a stage where we can really foresee that in a not too distant future, you will have scientists coming up with an RNA synthesizing itself, unable to evolve by itself in a test tube. Now, the ribosome is really the most complex machinery, but is essential for life at the present time. So how did it come to be? That is really quite a big question. So we think that the ribosome, in fact, is a kind of ancestral genome which has evolved by modular evolution, and in fact, which has within it the ability to synthesize protein, but has also another region that is domain 3 that we think is in fact a relic of the original replicase that was there for synthesizing the ribosome, or the RNA entering into the composition of the ribosome. So this is basically where my laboratory is essentially focusing on, with funding from NASA, but to tell you the truth, I really think that what we will need is really Catholic funding for being able to achieve this. And I think that if we can demonstrate that there's an RNA replicase buried within the ribosome, I think this is also a way to be truly witness of Catholic science within this area of research that I can tell you is very secular. So talking about life is also described by bios. So the diversity of all living organisms on Earth. So bios express the diversity of living organisms, and this is a tree of life as we can see it out of the analysis of the ribosomal RNA. Basically, a tree that has 3 major branches of life, the bacteria, the archaea, and the eukaryotes. And we are part of this branch of life. Now, as you can see, this is a classic tree, but the reality of the real evolution of this tree is that there has been quite a bit of endosymbiotic events that lead, in fact, to a mix-up of some bacterial cell with proto-eukaryotic cells, leading, in fact, to the modern type of eukaryotic cells we're all made of. What it means is that every cell that we have in our body, most of them, what they have are organelles that are directly related to bacteria. So we are a mix of bacteria on a proto-eukaryotic cell, emphasizing that without bacteria, we will not be there. And again, the reality of the tree is not merely to see some of these branching between branch of the tree. In fact, here is a better idea of the tree, a tree with a large trunk where all these branches basically could potentially lead to transfer some information between them to lead to the formation of the diversity of living organisms on planet Earth. What is interesting is that when we're looking at all the living organisms, we're looking at all the species of organisms, but it's also clear that many of these species could, in fact, sometimes create hybrids, and that is still taking place at the present time, and there's more and more evidence that hybrids could be formed between organisms that were initially thought to be species. So again, driving force for diversity and unity, really vertical and horizontal transfer of genetic information. The last point is about psyche. Psyche is also essential for describing life. This, in a minimalist type of idea, is emphasizing the cognitive life, the life as consciousness. Well, it's the soul, basically. But in a materialistic way, often reduced to mere consciousness, cognitive life. But this is an interesting concept because life, as we know it at the present time, shows this type of cognitive behavior at the smallest level of organization. And when we're talking about biology, it's not merely what we have learned in the classroom in the past. It's really now an idea that encompasses a larger vision that is basically encapsulated by the extended evolutionary synthesis idea of biology, which, by the way, takes advantage of the possibility also of having a context impacting the way the information at the level of a cell will be expressed. That is epigenetics. It's beyond what is within the gene. In fact, the context can basically imprint some message at the level of a genome so that the information could be treated differently by the cells. This leading to phenomenon of canalization, phenomenon that, in fact, direct precisely the evolution of some organism in some kind of direction. All this emphasizes that when we're talking about life, it's not merely bottom-up causal effect. It's not bottom-up causation alone. It's bottom-up, same level on top-down causation that takes place, which means that if you have molecular structural networks that could affect functional networks and cellular networks, you have also selection pressure that apply not at the level of the parts, but at the level of the whole, leading the cellular networks to affect the functional networks to affect molecular structural networks. This is basically bottom-up causation with top-down causation. And the reality of life is that it is a limit on constraints that are created by the whole that basically dictate also the information on functional cellular network, checking the quality of the information. So how the fundamental function of life emerge, that is a big question, but the reality still is that you need replication of DNA on RNA and the synthesis of peptide. So we have been proposing a definition for cellular life, that is to say that cellular life, the simplest form of life, is in fact an informational biosystem that enables this top-down causation by informational control or natural adaptive selection that takes advantage of Lamarckian mechanism. This basically showing that the whole is essential for the evolution of the parts. So when we're looking at life, through Psyche, bias, and Zoe concept, well, in fact, these concepts are unable to be dissociated from one another. They are nurturing the same substance, life. but having still some particular properties. Psyche relating more to the learning processes with memorization, bios to the way these processes are embodied at the level of a cell and individuated between a species, and Zoe being the principle, this principle that leads in fact to the replication or reproduction or metabolism of the cell. So when we contemplate with wonder the universe in all its grandeur and beauty, we must praise the whole Trinity, which is in fact reflected in bios, psyche, and Zoe. Psyche being the Father, reflection more the Father, bios of Christ, Zoe of the Holy Spirit. All this one in the Holy Trinity. My conclusion, what is life? Well, life is love incarnated. And this is basically a heart that we have been able to generate out of RNA. Out of a syntax of different RNA modules, we build up these nanostructures that totalize 800 nucleotides, and that as one strand of RNA is folding into a shape of a heart. Here are other hearts that were generated basically in the laboratory. The bottom line is that what we need to remember is that without love, nothing is created. In fact, only loves create. And my hope is that the science of the future, as a contemplative exercise, will basically be this science that will contribute to the civilization of love. So thank you for your attention.
We have time for one question. Kathy.
And this is going to be very basic because I don't know any chemistry. But early on in your presentation, you showed, I think it was Steve Miller in the 1950s, putting the elements together and creating the amino acid. But there was something added. There was a spark for the excitation.
Energy, yeah.
Where is that in the natural?
Oh, well, it's lightning.
It is always lightning.
Well, it's not always, but in an atmosphere, it could be lightning.
Okay.
So, yes.
What other things could it be?
Well, in interstellar space, in fact, you have UV light, you have UV radiation, you have plenty of radiations of different kind, and UV radiation are very effective at leading atoms to be linked to one another through covalent bonds.
Thank you.
Okay, quick question.
I believe reading a thought by Leslie Orgel on the polymerization of organic species that he says could occur in water that undergoes freezing, where there is a concentration of the organic matter as a result of the ice formation. Could you comment, please?
Yes. In many ways, I didn't exemplify too much the role of the context, but it's clear that context matters. And there's a phenomenon such as convection that could be taking place in some of these in liquid in connection with a microenvironment that could lead, in fact, to a phenomenon of concentration on freezing, sowing, freezing, sowing, freezing. Basically, alternating between day and night, could lead water to freeze, to unfreeze, and this basically contributing to enrichment and concentration of some of these compounds. Yes.